So thank you, Betty, for the introduction. So Park Fulham is not really that far away from Clearwater Bay, like California is a lot farther. So first I have to thank the organizer and Salt for Eggs for having me here this morning. And it's been absolutely a great fun morning so far, even before the alcohol. And after, you're gonna have to ask my wife and kids tomorrow. So, so let me get going. So you've got a light bulb. It's blown. So what do? You go take it out, get a new one, and put it back on. So what do you do if you are heartbroken? So you go get a drink, find a new date. But that's not exactly what I meant. I mean, what are you going to do if you are really heartbroken, literally? You've got a heart attack, myocardial infarction. So if you cut your skin, assuming that it's not terribly bad, you're going to heal on its own because of the presence of skin stem cells. But our other vital organs are just not as lucky. If you've got a blockage in your coronary artery, myocardial infarction, heart attack, your heart cells are gone, they're going to be gone forever. You're not going to get back to what you were. In any case, if anything, things are going to get worse and worse. Your doctors can manage and try to slow down the progression, but there are absolutely no cures or whatsoever. So this is pretty bad, and in fact, some of the patients even progress to heart failure. When it comes to the late stage, the last resort is to get an organ transplantation. So you have to wait for a donor. You have to worry about side effects that go with a lifelong immunosuppression. So it sounds pretty bad. So my question is, can we fix it early, stop the progression, much like replacing a light bulb? So this is indeed a huge problem with a rapidly expanding and aging population. And we know that the heart diseases are the top killer in the world, accounting for 1 in every 4 deaths, and in the US alone is costing at least $100 billion every year, and there are no cures. And what you might not know is that the heart is also the main reason why you are not getting quickly enough newer and more effective drugs and therapeutics. And I'm not talking about just for heart treatments, but a bunch of other conditions as well. For example, some pediatric patients, they get their cancer conditions taken care of, but some of them end up dying because of drug-induced cardiomyopathy. And Vioxx, an arthritis drug, has been shown to increase your risk for heart attacks, and therefore it was withdrawn after entering the market, leading to billions and billions of dollars of lawsuit settlement. And let's not even talk about the R&D costs associated with this original development. And the list goes on, and there are drugs that are originally meant for weight loss, inflammatory disease, gastrointestinal diseases, et cetera, that have been withdrawn from the market. So drug discovery, and this is another big problem. So in addition to human heart diseases, drug discovery and development has been traditionally an unbearably expensive and ineffective process. So let's say you're starting off with about 1,000 drug candidates. You do a bunch of standard laboratory tests, and you end up having about 50 lead compounds that may be worth going further, and you put them into different animal models for assessments, and if you're lucky, you get one lead compound that may be suitable for clinical trials, so you go through the entire process of phase 1, phase 2, phase 3. This is pretty much like a one-way street, and at the end, you get this binary answer of whether it's a yes or no. But the odd is not on your side, because 80% of the time, you fail. And the top reason has been toxicity to the heart, even for non-cardiac drugs. So it primarily has to do with a lot with the fact that animal models are inaccurate, so they don't pick up the side effect at this stage. And animals are animals. I know people can tell you that the fish genome, the mouse genome are very similar to the human genome, so at the end of the day, animals are animals. So if a mouse is smarter than a human, it's not a mouse problem. So so these are the shortcomings, and 1,000 candidates down to 1, and 80% of the time it fails, and we are talking about it takes about 10 years with a financial cost of about $4 billion altogether. So if this is not a broken process, what is? So can we do something about it? So clinical trials, the requirements are set by regulatory bodies, relatively fixed, so let's take this out of the equation. But what we can do is to shorten the preclinical time for drug development, reduce the preclinical costs, and even more importantly, improve the success rate of this. So instead of 80% of the time it fails, can we flip the ratio? And how do we do that? So a key thing to consider would be, I'm sorry that this is not really working the way that I think it should. Anyway, so here we go, sorry. So a key thing would be to identify the best candidate that we feel most confident with before entering clinical trials. So to make this process, to loop this process such that it's iterative and high throughput. So if animal models are not working that nicely, can we think about replacing it with, let's say, genuine human hearts or even human bodies, surrogates, before putting anything into humans? So that's what we are proposing to fix. So what is the solution? We think that we have a potential solution, and the video that I'm about to show you is only 2 minutes long, but it basically summarizes our 15 years of work. We started when I was at Johns Hopkins. Thank you. So all we have to do to start the process is to withdraw as little as 2.5 ml of blood from you. And you are looking at, in this particular case, mononucleated blood cells, and with a process called reprogramming, we can convert your 2.5 ml of blood cells into a very unique stem cell type called the induced pluripotent stem cell. I'm not going to get into the technical details, and I don't want to bore all of you with such an exciting morning. But the idea is that this is our starting material. So from now on, I'm going to be showing you technologies that we've developed in-house. So basically, we've come up with a way to instruct this stem cell type to become human ventricular heart cells. And here you're looking at one cell. Within days, we can mass produce and generate billions and billions of human heart cells that can function as fundamental building blocks. So these are really light Lego pieces that we can therefore put together as a range of different engineered tissues. Now think about if we, so by the way, this is not fairy dust. My children would tell you that magic is not real, but science is. So this is technology. So if we put about 1 million of these building blocks, Lego pieces, into a bioreactor that we design, within just a few days, we can show in front of you a beating, contracting human heart muscle, which is morphologically not different from what we can isolate surgically from your native heart. And imagine one day this is going to become a bandage for your myocardial infarction. Now, if you put more cells, like 10, 20 million cells, into another bioreactor that we kind of like design separately, within also just a few days, you can see a beating, contracting heart that belongs to you and is beating, contracting right in front of you. And you can make all sort of measurements that cardiologists do in the clinic, and it's personalized. It belongs to John Doe from the US, Jane Doe from Canada, and anywhere, anywhere else in the world. So I showed you the cartoon, and I'm going to show you the real thing here. You're looking at a beating, personalized heart contracting at 60, 80 beats per minute. And if you scare the heart by simulation, now it's beating 120 beats per minute, 180 beats per minute. So very much like what you see in the native heart here. This is a contracting human heart muscle fiber. Sorry, the video is not working. It's supposed to be contracting. But anyway, so this is what we call a heart in a jar, human heart in a jar. So with our heart in a jar and other related technologies, we think that we can function as human heart models outside the body for revolutionizing drug screening and drug discovery, so patients don't really have to take the drugs and be the guinea pigs. And they can be personalized, because all we have to do to start the process is to take only 2.5 ml of blood from you, and we never have to come back to you to collect another sample. That is providing kind of like an unlimited factory. And with such, we can therefore address genetic diversity and design doses of different doses for different ethnic groups, bringing us closer to precision medicine, meaning that two tablets 4 times a day, that kind of scenario may have been worked for Panadol and Tylenol, but we know that is clearly not optimized. Now we can take things really further to the next step. And you can imagine that these are also prototypes for future transplantation. And here we're looking at another heart in a jar with instruments that we have designed to make all sorts of measurements, pressure, volume, loop, and ECG, electrocardiogram, etc. We are thinking that we want to miniaturize everything so that we can improve the sensitivity and throughput and accuracy of our system. So how can we do better? How might we? So we are thinking that whether we can develop, let's say, robotics to automate the entire process of interrogating these human heart surrogates, such that we can find out their responses to different drugs, different conditions, programmed emotions, programmed exercises, blood pressure, etc. And so that we just have to sit back and look at our iPad and find out what one's pharmacological profiles are, like what drugs work for you and what don't. And the heart really is just a starting point. So we want to be able to link it up with other organ systems as well. In other words, we are trying to create mini-me. Mini-me. So you don't mind having blood tests. So how about having mini-me's before you take the medicine? This is what I call the so-called a traditional way of giving medicine, is that you have a condition, and that condition probably has a panel of drugs. You go to your cardiologist. They give you like drug A. If it doesn't work, you go for drug B, and then down the list until you find something that works. But unfortunately, in some cases, the side effects can be lethal. So why don't we try to have mini-me's? You test the side effects, you figure out what works for you and what don't. So that's the idea. And so this is just a starting point. And I'm a big believer of having banks of artificial organs in future. For now, we are thinking about using them as in vitro diagnostics, but ultimately, I believe that we will have critical organs sitting in incubators or being cryopreserved, and so that when you want to use them, they're around for you, and you don't have to wait for donors. So you know what? Steve Jobs used to tell us that this pocket is for iPod. I'm a Mac person, so I wouldn't argue with that. But he didn't really tell us anything about this pocket. So what is that for? It's a mini heart. And we are working on prototypes. All we have to do is start to get 2.5 meters left. Thank you very much, and I'll be happy to take any questions that you might have.
Okay.